So the title of this conference is How Captair Biosmart Can Make PCR amplification totally reliable. That's probably the first thing to understand. So PCR is an amazing application that gives a lot of information for research, forensic, medical investigation. But at the same time, that's an application that can be easily disturbed by contamination. So when I started to study 20 years ago this application, that's the first thing you learned about PCR. It was the case 20 years ago. Today it's a little bit more reliable, but what I learned 20 years ago is that PCR is very sensitive, and if you do not perform your application totally well, at the end you're not sure about the conclusion of your application. So that's totally amazing. Okay, so let's start with the definition of PCR. PCR means a polymerase chain reaction. Polymerase chain reaction, maybe for some of you, it's not totally clear, but the idea of this application is from one simple sample of RNA or DNA, you know, the code, the genetic codes you've got in your cell, from a small sample, to amplify it in order to create replication of this genetic code. So the application was discovered in the 80s. So there is a kind of debate to know how did create PCR. And it was also, there was a big fight about the patent in the 80s between Dupont and Moore and a researcher. So I don't want to come back to this original crisis. That's another story. And in 1992, we had the first real-time PCR by Iguchi. I think it was in Japan, if I'm correct. So the main problem we've got when you sample DNA or RNA in one cell, as example, you find her hair and you want to know where this hair is coming from. So you will extract the DNA in the air, but the quantity of DNA you've got at the beginning is not enough to conclude anything. And the first thing you've got to do before analyzing the DNA is to amplify it. And that's where PCR is very useful. Second part of the introduction, also very, I think, something to understand. I think most of you know DNA. That's what we called desoxyribonucleic acid. So I think nobody knows desoxyribonucleic acid, and everybody call it DNA. DNA is this fabulous double-strand helix molecules that embeds all the genetic codes of yourself, but also of all the animals, plants, bacteria, or virus, most of virus in the world. The genetic codes is very similar to the, an informatic codes with 0 and 1, but where the 0 and 1 are replaced by C, G, A, and T. And the sequences of C, G, A, and T will create what we call the genetic codes. C means cytosine, G, guanine, A, adenine. I don't know how to pronounce it. That's not the only word today. I don't know how to pronounce it, and the T means thymine. Okay, and what makes these molecules totally unique? You've got one strand with these sequences of C, G, A, T mixed all together to create one unique sentence of with 4 letters. And each strand is combined to another strand, which is exactly the image of the first one, knowing that each C of the first strand is combined or associated to a G of the second strand, or each adenine of the first strand is associated to a thymine of the second strand. So knowing that when you've got one strand, you can easily rebuild the image of the first one with, if you've got the accurate amount of cytosine, guanine, adenine, or thymine. And so that's what DNA is. RNA is a little bit different. It's also a genetic code. You can find it in combination with DNA in human cells or in plants, but also some organisms used RNA as their major or unique genetic codes. The difference is that in RNA, the genetic code is not made with a CGANT. There is no thymine in RNA. Oh, sorry, there is a mistake on the slide. The T is replaced by uranine, a U. And the fact that you've got uranine and that this is a ribonucleic acid and not a desoxyribonucleic acid means that the RNA cannot create this double helix as the DNA. That's a single strand. So now I will explain step by step how PCR works. So I just took the example of the reverse transcriptase PCR, but that's not very different of the normal PCR. The reason why I selected to explain reverse transcriptase PCR is because that's the one we're talking about when we talk about COVID-19. COVID-19 is a virus, and this virus, the genetic code of this virus is RNA. So the idea is that RNA cannot be combined to another strand of RNA. So what we use in reverse transcriptase PCR, we use an enzyme. I don't know, again, another word I don't know how to pronounce it, that is called reverse transcriptase. And thanks to this one, we will associate a single strand of DNA to the single strand of initial RNA. After that, we hit this association of RNA and DNA at about 94 or 96 Celsius degrees. It depends on the procedure. And in order to recover or to extract this single strand of DNA. And based on this single strand of DNA, as explained in the previous slide, we are able to create the image of this single strand, the combined image of the single strand in order to create a double helix or a double strand of DNA. So from one RNA, during the first cycle of PCR, from one RNA, we create a double strand of DNA. And based on that, we go to the second cycle. And again, we use the first double strand of the DNA. We denature it at 94, 96 Celsius degree in order to separate the two strands. And from the two strands, we are now able to create two double strands. And if we do it again during a second cycle, a third cycle, we denature at 94, 96 Celsius degree in order to create four double strands of DNA. And you can see here in 3 cycles, we create from one RNA 4 double strands of DNA. And that's exactly how PCR works. From one small sample of DNA or RNA, you will multiply it to a large amount of genetic codes. And that's very useful for research. There is different kinds of application of PCR. I think the most popular one is probably forensic investigation. If you find blood samples, hairs, or whatever, you can extract the DNA and analyze in order to identify the culprit of murder, as example. That's probably the most popular, but that's not the most useful. The most common one is the use of genetic amplification thanks to PCR to establish diagnostic or parentage as example. And that's exactly what is done for the COVID-19. We can also use PCR to identify microorganisms or use it for biological research. So, few words about COVID-19. That's just a reminder. We talk about RT-PCR, and that's a topic today. But we are at the time for most of northern countries. We are at the time where we say that after confinement, we've got to test more and more people, and we will use two different tests. The first one are the serologic test. Nothing, so that's got nothing to do with PCR. That's something that uses different methods, and this is used in order to find antibodies in blood samples. It does not tell you that you are sick regarding COVID-19. It just tells you that you were in contact with COVID-19 and you developed antibodies to fight against it. Now, the RT-PCR that we will talk about today is the amplification of genetic codes, and we track the virus into nasopharyngeal samples. The order is to extract it in your nose, to extract and to find the presence of virus in your body. And we will do that by amplifying the genetic codes of the virus thanks to PCR. So, as said at the beginning of this conference, so this application is very, very sensitive to contamination. And if your sample is contaminating during the first phase, the pre-PCR or during amplification, the first risk is the overcost. And just as an example, here in Europe, we estimate that the cost of one PCR for COVID-19 is 135 euro. So if the result is bad, that's a waste of money. But not only, because a wrong diagnostic can lead to bad medical treatments, and that can be very, very bad for the patient. Also, that's also something that is clearly understood by most of people, is that if for forensic investigation, it can lead to judicial error, or for research, wrong research interpretation. So a bad PCR with contamination can lead to very, very bad results and bad consequences. Again, we've got to understand that the contamination can occur at different steps. And for that reason, when you perform PCR, that's very, very important to differentiate three different steps that must be done on three different locations. The first step is extraction. At the beginning, I told you if you investigate, as example, if you're looking for the presence of virus into your nose, first you've got to sample to obtain the nasopharyngeal samples. And unfortunately, the RNA you're looking for is embedded into your sample. And if you just consider the virus, the RNA is embedded in a capsule, and if you just use the capsule itself, you cannot amplify it. And the first thing you've got to do is to remove the RNA from the virus. And for that, you've got to break all the walls of the capsule in order to remove the RNA from the cell. So when you sample virus, it's a pathogenic cell. So in that case, you've got to do it in a BSC workstation type 2 according to NSF 49 or according to EN 12468 69, sorry. So extraction, if the sample is pathogenic, must be done in PCR. If it's not, it can be done in any other workstation. That said, when the RNA or the DNA is extracted from the sample, it's no longer pathogenic. And at this step, it's very important to move it to another workstation that will be dedicated to the pre-PCR in order to avoid the contamination of extracted RNA by new samples. So the pre-PCR goals, workstation's goals, is it's a place where you will mix your DNA or RNA samples with primers, with, sorry for the word, nucleobases, ACGT, that will be used in order to rebuild new DNA fragments, and with the enzyme, as example, the reverse transcriptase, if you want to amplify RNA. So this tape is very, very sensitive to contamination. And at this step, you've got to protect your sample against two major contaminations, the one coming from the environment and the one coming from other samples. At the end of pre-PCR, you cap your sample, you close it in a container, and you've got to move it to the PCR workstation itself. And amplification is performed in the thermocycler, and the thermocycler only use capped containers. Normally, it's not highly sensitive to contamination. So the most sensitive part is the pre-PCR. So the challenge is that between the beginning of the application, where you've got a very, very small amount of DNA or RNA, and the end of the application after amplification, you can have 1 billion strand of DNA at the end. So the ratio of quantities between the end of the application and the beginning of the application is huge. And it makes your sample at the beginning very sensitive to contamination. So this contamination can come from other samples, that's what we call cross-contamination, and it can come from the environment. That's what we call environmental contamination. And a good PCR workstation must protect both against cross-contamination and environmental contamination. And that's what we expect from Captair BioSmart PCR workstations. So that's what I will explain now. First of all, when you introduce your samples in the PCR workstation and when you start to perform your pre-PCR, you've got to protect your samples against environmental contamination. And that's done by containment of this workstation in a vented enclosure where the air is pumped into the room, filtered on high efficiency filters, and the dust-free air is pushed to the worktop. So thanks to this principle, all the air that is in contact with the sample is dust-free, and I will explain how much it's dust-free. But also, thanks to this ventilation, you create an air barrier between the user and the application, and this air barrier avoid any contamination from the user to the samples. The filtration is performed thanks to two types of filters. So a standard H14 filter according to EN 1822 part 1. So H14 has got an efficiency equal to 99.995%, but also it can be equipped with filters with higher efficiency. As example, U16 filters, 99.99995% of efficiency. And on the Captair BioSmart, we've got also the ability to combine it with molecular filters, because recent research has shown that VOCs can contaminate your PCR during pre-PCR. So the first thing to understand that you've got different type of filters. The ones that are embedded into the BioSmart are specially designed for this BioSmart workstation, and they have got different properties. The first one, so they reach the requirements of EN 1822, but also the ones that are used by ERLAB have got a very low pressure drop. And this low pressure drop enlarge the lifetime of the filters, and average filters lifetime of these filters in PCR workstation is around 3 years, between 3 years and 5 years. But also it guarantees a lower energy consumption, and something that is also very important for the end user, it's less noise. So people will perform pre-PCR in some laboratory will spend hours in front of their workstation, and that's very important for them not to be annoyed by the noise of the ventilation. The other things we want to promote is that the size of the filters is exactly equal to the size of the worktop, and that's also very, very important. The efficiency, not the efficiency, but the airflow that will pass through the filter is monitored in real time thanks to an embedded anemometer that guarantees both that there is no clogging of the filter and that there is no need to change it, and also that the front velocity ensures no contamination from the user to the sample. And the molecular filters that can be combined, optional molecular filters that can be combined to these HEPA filters, are totally compatible. They've got the same contact time, same airflow performance, and they are dedicated to VOC trapping, and they can be combined to optional molecular S sensor. The molecular S sensor is a sensor that is focused to detect VOCs into the atmosphere. A very common question coming from users in the world is, are you sure that HEPA filters can trap virus or bacteria? So I will try to explain, but according to EN 1822, filters are tested following a method that is called MPPS. It's most permeable particle size. So it's the size of particles that is the most difficult to trap. Common sense tells you that the most difficult to trap is the smallest one. But when you try to investigate, you quickly realize that that's not the case. That's the case, but up to a certain level. So, and this level is between 0.15 microns and 0.3 microns. And below or over this value, efficiency of filters is higher than the minimum efficiency that is guaranteed by EN 1822. That said, so it means that if you buy a filter that is does comply to EN 1822 and that is H14, it means that its efficiency, guaranteed efficiency, 99.995% for the particles that are the most difficult to trap. But all the particles that are smaller or bigger than these particles are trapped with a better efficiency. So again, I give you on this slide some values of the different size for bacteria or virus. For bacteria, the smallest bacteria have got size around 0.2 microns, and you can see, according to the MPPS test, that this is totally compatible with HEPA filters. Virus are smaller. The smallest virus have got size around 22 nanometers. But again, knowing that below the size of particles that is the most difficult to trap, the efficiency increase, we can also show that this is very well trapped by HEPA filters, just considering the size of virus. But we've got also to understand that viruses are never totally free into the atmosphere. Most of the time, they are linked to small vesicles or small particles into the atmosphere that are totally trapped by HEPA filters. Another very important question and very common question coming from customer is that how can we evaluate the cleanliness of the atmosphere into the enclosure? For that, we can use different standards, and the most commonly used is the ISO 14644 standard. That is written to describe clean rooms and associated controlled environment. We can say that a Captair BioSmart is an associated controlled environment. In the ISO 14644, the idea is to sample the air into a room or into a workstation thanks to a particles counter, and to compare the results of your particles counting to this chart. And here, as example, you can see different classes of air cleanliness from ISO 1 to ISO 9. And as example, if I select ISO 5, when I compare my results of particles counting, I must ensure that there is not more than 100,000 particles of diameter higher than 0.1 micron per cubic meters of atmosphere. And at the same time, I must not have more than 3,522 particles of diameter higher than 0.5 microns per cubic meters of air, of sampled air. So that means that depending on your particles counting, you can reach certain level of cleanliness. Also, what we've got to understand is that the results not only depend on filters efficiency, it also depends on the environment. So to obtain a very good cleanliness, you need a very efficient filter, a very good vented enclosure, but at the same time, the environment where the enclosure is installed must not be too polluted. That's very, very important to understand that. The other important information is that the most commonly used standard is the ISO 14644. But in the world, you can find customers referring to other standards. And one very popular standard that is less and less used in the world, but still present, is the US Federal Standard 209. And on this slide, I I give you some equivalence between this American standard and the ISO standard. And as example, an ISO 5, according to the 14644, is almost equal to a class 100 according to the federal standard 209. 100 according to this standard means maximum 100 particles of 0.5 microns per cubic feet of air. You can also compare the result of ISO 14644 according to the good manufacturing practices that are used in pharmaceutical industry, and an ISO 5 is equal to a class A or B according to the GMP, knowing that the differences between A or B classes depend if your room is tested under activity or is in standby mode when you test it. So we can also ask ourselves what is the cleanliness we're looking for PCR. The most commonly requested air cleanliness for PCR, but that's also the same for BSC, the internal environment of BSC, is ISO 5. And here you can see that ISO 5 is also what is the most commonly used in pharmaceutical industry, in surgery rooms, as example. For better cleanliness, sometimes you find it for microelectronic because it's very sensitive. Light is very sensitive to dust, and lower cleanliness, you can find them for in agri-food industry or spatial industry or painting cabins, as example. Now, at the end of your pre-PCR, after you mixed your sample of RNA, DNA, plus primers, plus nucleobases, and before you, and after you chip it to the thermocycler, between two different applications, you've got to clean your enclosure to be sure that the previous prepared samples do not contaminate your new samples. And that can be done thanks to two technologies. The first one is the usage of UV lights. And the UV lights that is useful for the decontamination is what we call UVC or germicidal UV. When you talk about UVC, it means that the wavelength is centered on 254 nanometers. It's also very important to be cautious about the wavelength of your UVC bulb, because you must not generate ozone, and ozone is generated at 185 nanometer rays. So it means that your provider of PCR workstation must guarantee you no release of rays at 185 nanometers. And so to come back to UVC, UVC waves at 254 nanometers affect RNA and DNA and degrade them. So it means that after the destruction of DNA or RNA, thanks to UVC, you're sure that it cannot be amplified again. So, it's important to use the accurate wavelengths, but at the same time, you must use the accurate wavelengths at the accurate dose. And what we call dose is intensity, the intensity of UVC that is linked to the amount of wavelength of light that is generated on your worktop. So it depends on the UV lamp, the distance between the UV lamp and the worktop, and the way it's also distributed into into the chamber, into the enclosure. And it's also a function of times. And that's the reason why all Captair BioSmart are equipped with a timer that allows the user to adjust decontamination time. I told you that there is another way to decontaminate the internal volume of the enclosure. That's thanks to cleaners. I just want to focus that if you use cleaners, you must be very careful about the type of cleaners because they must not degrade the material of the enclosure. As example, if you think about stainless steel worktop, they can be damaged by certain type of cleaners. So if you want to know what type of cleaners to use, I highly recommend to contact your provider. So that to come back to UVC lighting, then you've got to determine how long you will decontaminate thanks to UVC. So the idea is that in order to determine the accurate time to decontaminate your enclosure, you've got to refer to the literature. And here, as example, I gave two examples. One for RNA viruses. I found this reference where David Little and José Luis Sacripanti determined that you need 12 millijoules per square centimeter of worktop to deactivate this type of virus. And below that, you've got one example for Escherichia coli. So depending on the destruction rate you're looking for, I gave you different amounts of UV that is needed and the time that is necessary to deactivate the DNA or of Escherichia coli thanks to the UV lamp. Knowing that here, that's an example for Captair Bio 321 with an intensity of light of 0.08 millijoule per centimeter per second. What you can see here, the timer, the UV timer of the Captair BioSmart can be set between 5 minutes and 30 minutes. And according to these two references, you can see that most of the time, 5 minutes of insulation, thanks to the UV lamp, is enough to decontaminate your enclosure. So Captair BioSmart, they are totally designed for pre-PCR, thanks to the ventilation when it's needed, and the ability to decontaminate with accurate lamp at 244, 54 nanometers. I just want to focus on the idea that these enclosures are not designed for work with pathogenic organisms, but when DNA is extracted from the virus, it's no longer pathogenic and there is no risk to handle it into a PCR. So that's very important to understand that. Other features, so all Captair BioSmart are designed in order to improve the cleanability, and the two worktops, the one in stainless steel or the one in Trespa Top Lab Plus, are manufactured in order to have no square corner. You can sweep it very easily in order to avoid any accumulation of dust or contamination in the corners. The front door and the post are manufactured with 10 millimeters UV resistance PMMA. And the assembly is optimized in order to avoid any leakage of UV out of the enclosure, because you know that UV is very dangerous for the user. So, and the front door is equipped with a switch. When you close it, so you're sure to avoid any UV leakage, and you can turn on the light with no risk for the user. All Captair BioSmarts are equipped with a slanted front door for a better ergonomics. I talked about the noise before. The front doors is another part of the ergonomic, and I can also talk about the internal LED lighting. So we designed this enclosure to be totally the ergonomics for the user who will spend a lot of time per day in front of the enclosure. The choice of the PMMA of the walls, polymethyl methacrylates that you use for the slanted doors is also selected because 10 millimeter thickness can stop beta rays, and sometimes DNA or RNA are marked with radioactive phosphor at low dose, and that's so the thickness of these PMMA panels is selected to be sure that there is no escape of these rays out of the enclosure. Each unit is equipped with 2 modes, one UV decontamination and handling. So you've got a cycle between the time where you decontaminate, everything is closed, there is no sample in it, just to be sure that when you will start the handling, everything is decontaminated. And during the handling, so the unit is vented or not. We'll see that we've got different option for that, but you must be sure that there is no decontamination thanks to these 2 modes. They are equipped with high efficiency UV bulbs centered on 254 nanometers with no risk of emission of ozone, and that's also very important for end user. And the internal LED lighting generate daylight lightnings at 900 lux. The units are simple to operate. We want the user to focus on their application, and it's also very easy to assemble or to maintain by yourself if needed. So we've got 3 different types of bio. 2 BioSmarts, the 321 and the 391. The 391 is a little bit larger. It's a 1 meter large enclosure, so that might be a little bit more comfortable if you've got more samples. But also the internal insulation, UV insulation, is a little bit higher than on the Captair BioSmart 321. Both units, the 321 and the 391, are both equipped with UV lamps and HEPA filtered or optional HEPA filtered to ensure air cleanliness into the enclosure. We also sell what we call the Captair Bio 320. This enclosure is not vented. It's designed to be installed in clean rooms where all the room is already dust-free. In that case, you do not need to have any HEPA filters on your worktop, but it's equipped with high-efficiency UV lamps, the same than the one used in the 321, and that's got almost the same dimension. So to conclude, that's very important to understand that PCR applications are very sensitive to contamination, and from the extraction to the amplification, that's very important to be well organized with 3 different workplaces. One for the extraction, and if the sample is pathogenic, it's important to do it in a BSC. The second one, where you mix your sample with primers, with nucleobases, and what we call pre-PCR, must be done in a pre-PCR workstation. Thanks, as example, the Captair BioSmart 321, 391, or 320. And then the amplification itself must be done in thermocycler, and that's also another place. And when you select a PCR workstation, it's also very important to ask your provider to guarantee a perfect UVC insulation thanks to the accurate UV lamp with no release of ozone. So thank you for your attention. Now it's time for question. As explained by Mathieu before, if you've got any question, you can use the hand.
Yes, thank you, Dr. for the presentation. If you have some question, you can use, as I said, the little hand on the right, and I will open your mic. During the conference, we had a question from Juan Manuel Sanchez. The question is, because the ozone is a germicide, where is the problem of generate ozone in the hood using UV at 185 nanometers? This could enhance the cleanliness inside the hood.
It's totally true that ozone is germicide and can destroy microorganisms, but at the same time, it's very, very noxious for the users. So knowing that if you use a correct UV lamp, the UV lamp itself is very, and UV wavelengths are efficient to destroy the virus. That's better not to generate ozone that can damage or contaminate the user itself. So each time you can avoid a generation of ozone is better.
Question from Victor Debas.
Yeah, can you explain to me what a dead air box is and what's the purpose for it?
The 320.
Yeah, I've seen it being spec'd here and there, but I couldn't understand. I assume it's a PCR cabinet without any ventilation.
Yeah, correct. So the first two the 321 and the 391 are both vented, and it guarantees you the air cleanliness and the UV insulation. But we also noticed that some users use this unit in clean rooms where all the room is already vented with clean air. And in that case, you do not need to protect your sample against environmental contamination because you already work in a clean room. And so it means that the 320 with no ventilation, it's just used to avoid cross-contamination between two series of pre-PCR.
Okay. So in other words, your room has to be sterile in a way for it to be. Okay.
Yeah.
Okay. All right. Thank you.
You're welcome.
There is a question from Josette Chan. What are the difference between the Captair flow and bio?
The answer is complicated because it depends where you are. We do not sell exactly the same range of products in Asia and in Europe. So first part of the answer is the situation in Europe. In Captair BioCap, you've got both UV lamp and ventilation, or no ventilation, but you've got always UV lamps in bio, and they are 100% designed for PCR. So the UV lamp, the distance between the UV lamp and the worktop is optimized for pre-PCR, and everything is optimized for pre-PCR. And in the flow, you've got no UV bulb, no UV insulation, and I told you that this is only in Europe. The thickness of the PMMA panels in the flow in Europe is only 8 millimeters, and there is this vented workstation are used to produce dust-free space for typical biological application, electronic, cell culture when the cells are not pathogenic or things like that. That's what is done in Europe. And in Asia, we've got some flows are also equipped with UV lamps. I would say probably depending if you use marked phosphor or not in Asia, you can select between bio or flow depending on the size or depending on your applications. They are more similar in Asia than in Europe. The only difference is the thickness of the PMMA panels on the bio that is 10 millimeters instead of 8 in Asia. And the internal height of the enclosure also is different.
Question from Biranda Mila. Which is better to use BSC or Captair Bio?
There is nothing better than the other one, so it depends on the situation. First one, clearly, I highly recommend to have, if the sample is pathogenic, I highly recommend to have both. The BSC for the extraction, where the pathogenic cells are, and the pre-PCR workstation, the bio, for the pre-PCR. As the virus is no longer pathogenic when the DNA is extracted, there is no risk of contamination for the end user, and it's more ergonomic, it's cleaner and safer to make the pre-PCR in the bio. If the sample is not pathogenic at all, there is no need of BSC at all. So it really depends on the situation.